Can you please confirm if you can see my screen and I'm audible enough?
Yes, sir. Screen is visible and you're also audible, sir.
Okay, thank you. Welcome, everyone. And we are going to discuss the next chapter or section of the syllabus that is about vectors. As I already described, and I think I gave you good enough introduction to vectors somewhere in the previous discussions. A vector is something that carries. In biology, that's how we use in infectious diseases. Several organisms may be like, there may be as vectors which will say for Plasmodium malaria, mosquito carries the vector, the Plasmodium from other sources, one human to the others. And not just that it carries, but there is also a reproductive cycle kind of thing within, for Plasmodium within the mosquito. So here in recombinant DNA technology, we are interested about a gene of interest, right? Or a fragment or a DNA molecule of interest. DNA, or actually we should speak of, every time we speak of DNA, we should speak of it as information, right? DNA is one of the best molecules ever, because every molecule, you can take any molecule. Every molecule is a physical substance, and every molecule has some occupancy of space. That means they have a design, they have a shape, they have properties. You can call that as information, right? You can take any molecule and everything is information basically. And the thing that we are discussing here, every molecule has, chemically wise, everything is a physical thing, and it is composed of information depending on how we perceive it. There is only one molecule that we know of, that those are nucleic acids. That is a physical substance. It has information. It has inheritable information. RNA. And that inheritable information is because of base pairing. So information here for us, it would mean the sequence. Sequence has properties. Different sequences have different properties. Nucleic acids and different sequence of amino acids in a protein also has different properties. So,
Sir?
okay. Internet connection is lost.
Sir, could you hear me, sir? Sir?
Yes, I can hear you now. I understand that a network problem, or you had a question?
Sir, this network problem, sir. Like you were writing something.
Yes, yes.
Like you were writing physical, those things, and then you were describing that information.
I just started physical.
Yes, sir. Like that physical and information, those things you were explaining regarding that inheritance, and all of a sudden, writing that it was stuck.
Okay, I'm trying to use my mobile internet. Okay. Yes.
So now it's, sir. Now it's audible, sir.
Okay. Can you see my screen?
Yes, sir. Yes, sir. Okay. Like you were describing that inheritable information, and then sequence somewhere. I think there you were describing, sir, and then all of a sudden, like the pointer using that eraser, the pointer was stuck in just that one position, and then
Okay.
I didn't know whether
That's
you
okay.
were writing anything else afterwards. So
No,
that's
I
why.
did not. So, thank
Okay.
you.
Yes, sir. Okay.
So I think I was here. Thanks, Ajit. So when we are cloning anything or we are studying about something, it is actually information. If you take molecular genetics, you can call it as bioinformation, right? Information system of the living systems. Sequences in nucleic acids has certain properties. Sequences in proteins also has some properties. That is where enzymes, structural proteins, everything come about. In biological system, sequence is everything. So you change sequence, that means the information has changed and the properties will change. So often when we are trying to learn about it or manipulate or use it, employ it for some purposes, we clone the DNA. Or we try to, if we want to study the information, the consequences of it, then also we have to clone and characterize. So for this, the important thing here is the vector. This all region is important, without which it becomes difficult. It's not impossible with the current day technology, but it becomes little difficult. So we will have a discussion on those, and things could get little complicated at times. So step back, and sometimes you may have to visit your fundamentals in molecular genetics, and probably you can figure it out. But of course, you're always welcome to discuss with friends and me as well. Just post the question, and then we will have a discussion. In the last class, we also discussed about how to increase the recombinant DNA. That is, when we perform ligation between gene of interest and that of the vector, say this is the vector, and this is BAM-H1 site, and if you are having BAM-H1 sites here too, and we add ligase. This is what I'm trying to do right now is review what we have already discussed previously. When we add ligase, there are several possibilities. Again, I'm trying to describe how you are supposed to think about when it comes to this vector selection and so on. In this case, of course, there is restriction digestion and then ligation. Okay. So one of the possibilities is the recombinant DNA. That is what we are looking for. But there are also other possibilities where you may have multimers of the gene of interest itself binding like this. There are also possibilities of the vector alone religation. You call that religation. And as more and more of these components are consumed in the formation of fragment-fragment ligation or vector-vector religation, the probability of recombinant formation would reduce. So if we want to increase the probability of recombinant formation, we should somehow ensure that we should reduce the formation of vector-vector or fragment-fragment or vector-vector religation. To do that, we have discussed about, can anybody say the enzyme that we said is useful in this? How to increase the formation of recombinant DNA in this particular instance of what I am describing right now? Yes? The vector should have been treated with alkaline phosphatase.
That's alkaline phosphatase.
That's right. Thanks. Alkaline phosphatase, right? Then you're removing, what does alkaline phosphatase do? It removes the 5' phosphate. So, and there are other things we also have discussed about directional cloning. And there are, in this, I'll quickly go by. So all open reading frames have a direction. That is, They start with the start codon and they stop with the stop codon. It could be TGA, TAA, or TAG, either any of them. So when we want expression from it, transcription would be in such a way that the promoter is always upstream of the 5' side of the open reading frame, right? Something like this. So using this promoter, the RNA polymerase will make the mRNA. From mRNA, ribosomes will make the proteins. If it is cloned in a different direction, then the transcription would have been the antisense of it. This is the sense. You call it as mRNA as sense. You can call it as plus strand. You can also say it as coding strand. So if we have the open reading frame in this way, then what we will get is the antisense of mRNA. Antisense of mRNA, and there could be a protein probably if there is a open reading frame on the antisense RNA, but otherwise there will not be a protein. Right. First of all, we should also remember that there should be SD sequence if you're talking about, or ribosome binding site. When the mRNA is produced, the mRNA should have the ribosome binding site, which is called a Shine-Dalgarno sequence in prokaryotes or Kozak sequence in eukaryotes. Of course, there are differences in the way mRNA are recognized in prokaryotes and eukaryotes. So when we are cloning, we should ensure that we clone in such a way that the vector has the promoter. And if that is the case, we should be able to put the open reading frame in such a way so that the sense mRNA is produced and the protein is expressed. To do that, we try to employ different restriction sites. Unlike in here, in the first case, we said BAMH1 site, BAMH1 site, and also in the vector BAMH1 site. In the directional cloning, typically we use 2 different restriction sites, okay? And for that, we may use BAMH1 and EcoR1, SAL1, KPN1, any of those, depending on what is available. And we also discussed about designing primers in the last class too. I encourage you to have a look at that before. And we discussed some PCR-based and also some PCR-based methods to find out genetic variations. We will repeat that at some other point too. But I want you to look at design primers, and I hope you are aware that CIA should be conducted soon, and all the things that will be taught until this Saturday will be a part of CAA 1. Okay, please tell your friends, those who are absent.
So then when is the exam, sir?
I will discuss with you people, and we will decide, because I have to ensure that there is only one exam for you and so on. Okay, and you can also give us your favorable times in the WhatsApp group. You discuss among yourselves and tell, because there are other bioengineering MNT also, and then we can figure out. We'll discuss about it later.
Okay, sir.
Okay. So what are the properties required? As we discussed some time ago, we say the first thing, any DNA molecule to qualify as genetic information should have an origin of replication. Origin of replication is very important. If it is not there, there is no scope for this DNA to be replicated. And if it is not replicated, it is not inherited. Okay, so first thing we should say is every vector should have an origin of replication. And if you have a DNA like this is the gene of interest, and we put it directly into a cell, and this may produce something, okay, but eventually it is going to be lost or degraded. right? Why is that? Because there is no origin of replication to this fragment. If it already had an origin of replication and its ends or it is circular, then it is a vector. You can say it is a replicon, you can say it. So second important thing is we should have some selectable marker. That means how can we select the cells that have the vector and from those that do not have the vector. So selectable marker. That is usually an antibiotic resistance gene. Resistance gene, or there are also other ways which can give color, which can be of biosynthetic genes and so on. And we should be able to cut this DNA. Well, why we are discussing about all these things about vector is that we want to put the gene of interest or DNA of interest into it. That means we should have some way of cutting the DNA and say, for example, damage1. And these sites should be unique. When I say unique, it means that damage1 should not be somewhere else. So the restriction site should be there and they should be unique. That means there should be been only one of those restriction sites within the whole of the vector. And is it good to have one or multiple? If you have more number of them, more number of restriction sites like BAMH1, EcoR1, KPN1, and others, that is much better because then you will have flexibility. It's not just only the fragments that have BAMH1 that can be cloned in this vector, but maybe if you have those with EcoR1 can also be you cloned into this vector. That means it increases the probability of types of different sequences that can be used, that this vector can be used for. So these are called as multiple cloning sites. Multiple cloning sites, and those are restriction sites, which should be unique. Yes, anybody has question? I can't see the screen, so you can please tell me.
No, sir, I said it's MCSR, sir, that I mentioned it as polylinkers.
Okay, yeah, that is also fine. So, yes, I hope you understood these. These are the three essential ones. And some of the vectors will have even multiple ones, multiple selectable markers too. They may have more than one. Some of them may have more than one type of origin of replication. What you should remember is the origin of replication is highly specific. That means the origin of replication of E. coli is different from origin of replication of Mycobacterium, okay, or Bacillus, for example. So origin of replication will determine what can grow in a type of particular host or not. Host here is host cell. Like most often what you'll see is E. coli is the most commonly used for all manipulations. Majority of the manipulations, E. coli is commonly used in every lab. So origin of replication is specific to the host cell. When I say that, it means what type of proteins recognize this origin of replication. The host cell should be able to recognize the host cell, this origin of replication, and then initiate. So if I want a vector that can be cloned in E. coli, that can be replicated in E. coli, but also replicated in, say, for example, Mycobacterium tuberculosis, then it should have two different origin of replications. Okay, it all depends. The basic needs, of course, are what I have already elaborated. There should be at least an origin of replication that is specific to the host cell. There should be at least one selectable marker so that we can figure out those cells that have the vector and those that do not have. The third important thing is they should have cloning sites, unique cloning sites. Multiple is always better, so that you can use it for multiple purposes, to clone multiple or different types of genes of interest. Okay, and we will be getting into some more things before that. Okay, let's get along and then we will see. So this is from the textbook, and here is a vector, and this is the origin of replication. There is BAMH1 site, and BLA is for beta lactamase. Beta lactamase is an enzyme that degrades the lactam ring present in penicillin or ampicillin and other things. So by breaking that the lactam ring, it will provide resistance. It will kind of degrade the antibiotic. So it will be, the cell will be protected against antibiotic. Let's try to have a look at what would happen generally, how we would do it. We will take the vector, we will cleave it with BAMH1, and we'll also have a fragment with BAMH1, and then we will mix them and add ligase, often T4 DNA ligase. So what are the probabilities? You may have BAMH1, the fragment, multimers of fragments, because the ends, BAMH1 is compatible with another BAMH1. What may also happen is that vector-vector religation may also happen, right? If I put this into, now the next step would be transformation. And remember, this is the only recombinant one we are looking for, interested, that the researcher is interested. We will perform transformation, which means that by using chemical methods, we can induce the bacterial E. coli to take up the DNA from an outside transformation. And once you have, you say, for example, you will take about 1,000 E. coli cells, and you try to do transformation. Not all the cells will get the DNA. You can have different types. Majority of them do not get any DNA. Those you will call them as non-transformants. Okay, non-transformants. There will be a few, there will be a lot of them which may get vector-vector religation, like these ones. Assume they are about, say, 80. I'm just giving random numbers. We may also have fragment-fragment religation, which may be 5 or 10. Okay, that is, these are just illustration purposes. Don't take it really. Recombinant. So in here, if you see, all these are transformants. All these are transformants because they got the DNA. And once you have all these types of cells, and you plate them on nutrient agar plate or LB agar plate with ampicillin, okay, the non-transformants will die. Why will they die? Because they don't have any resistance to the ampicillin. What will happen of the transformants? In the transformants, fragment transformants will also die because they do not have the gene, the beta lactamase. Recombinants and vectors will survive because they have the beta lactamase gene. Where did they get the beta lactamase gene? They got it from the vector, right? The vector has the beta lactamase gene, or the antibiotic resistance against ampicillin. So what I'm trying to say is, here the purpose is that you get used to the terms. Non-transformants are those host cells that did not get any DNA from outside. Those are non-transformants. Although I have put this is not often any case because they will not grow on the colonies, on the agar plates. So what we now have are vector-vector religation, which I would refer to as a non-recombinant. These are non-recombinants because what was vector has just religated. There is no recombination there. Even if it is there, assume that there is no, that is not of our interest. Typically, you can call them as non-recombinants. And the one that we are looking for are the recombinants, which will have vector plus a fragment. Okay, please get used to the terminology. Everything is in the word. All of biology is probably 40% or 50% of it is in terms. If you know the terms and you know the meaning of it, you know 50% of biology, at least. So now we are going to discuss something about blue-white screening, which is interesting. You must have already read biotechnology people. And in this one has two things about it. First thing is we should know about what is meant by alpha complementation, and then blue-white screening. Okay. I hope you know the lacoperon, and we are not going to discuss all of it, but there is a gene LAC-Z, and you have the LAC promoter, operator, and so on. LAC-Z will produce an mRNA, and then upon translation, it will produce an enzyme, which is called as beta galactosidase.
Beta galactosidase.
That's right. Yes, beta galactosidase. And what it does is it is involved in breaking lactose to glucose and galactose. What it can also do is if we add XGAL, beta galactosidase will convert it into a blue compound. So XGAL is almost colorless, and upon treatment upon action of beta galactosidase, it will produce a blue color compound. Okay, that is one way of how to distinguish between, as I said before, you can have two markers if required in one of the properties of vectors. And here we are trying to see that, but let's get some biology correct before going further. There is a mutant of strain. There is a mutant of LAC-Z, which on its genome, the host cell, one of the E. coli cell, has a deficiency. It has LAC-Z prime, LAC-Z mutant. Okay, I'll put it within the star. It is on the host. This is on the genome. Okay. What happened was about this part of the DNA has mutation. It lost it. So it's not a complete open reading frame. If it produces, it will produce something like this. Only half an enzyme, you can think of it. And that half an enzyme is not so functional. Non-functional. So it lost this part, the 5' side, because of which the N terminus of the enzyme is not formed. The rest of the seed side, it is there. I hope you always DNA, or nucleic acids, we will always write 5' to 3', and proteins are written N to C, amino terminal to carboxyl terminal. So using some genetic studies, people have discovered that if we clone just the front, a small fragment, which you call it as alpha part. If we produce this one in a cell containing the rest of the part like this, then this one produces, this alpha part will produce this much, and from the chromosomal gene you will produce this, and now it forms a functional or active beta-galactosidase enzyme. Okay, so I will repeat once again what I was trying to explain here. So LAGZ is there. It's a large gene, of course, and they found some mutations in some of the bacterial strains, E. coli strains, which has lost this part of the gene. Although it is expressed, it doesn't produce a functional protein, right? Enzyme. It is only half of the enzyme. You can think of it like that. If we have cloned, or if we put in a different location, if you put in the missing part, and it cloned for the alpha component, and the host, say, I'll try to put it in a little different way. Say this is an E. coli cell. This is the genome, and where there is promoter, and this is supposed to be the beta-gal, but this is the only one that is present. The other part is mutated or lost. If we supply the alpha component somewhere else, right? The alpha component. So what will happen now is this will produce half the enzyme, something like that. And from this region, you get another half, the alpha component. These two can fuse and form a functional protein. And then they can convert X-gal, or they can even break galactose. X-gal can give rise to blue color. Okay, so this is about alpha complementation. There will be other details too about names and if strains and all. You don't have to really mug it up, but it's essential that you know the concept of it. Okay, so what we will do right now is we will try to go through blue-white screening. And PUC 18, usually plasmids are written like this with a small p followed by 2 or 3 alphabets representing whatever the lab or the regulation. It depends on the scientist who names them. Here it is University of California, UC. And number is also given. It's based on the series, and it again depends on the lab. There is one origin of replication, BLA, that is beta-lactamase. Do not confuse between beta-galactosidase and beta-lactamase. And then we have LAGZ prime, that is the alpha component, like I was trying to put this one. There is a selectable marker, right? One is already beta-lactamase, and then we are also having the alpha component of LAGZ prime. When you say LAGZ, it means full gene. When you say LAGZ prime like that, it means it is a truncated version or a mutated version. And people have engineered a multiple cloning site, which you call it as MCS. And I think I have a picture of it, how tightly they packed. Maybe I will try to get it for the next class. They have designed the multiple cloning site in such a way that all the restriction sites are very closely packed. And it is also a way of thinking. And using this vector, we can use any of the restriction endonuclease, say, for example, BAMH1, and then you cloned it here. You can ligate the fragment of interest. Let's go through the cloning procedure now. I hope you understood the alpha complementation part. So step by step, if we say, I'm not going to draw all the all the things. I'm only going to draw about LAC-Z and cloning site. Okay. So there is BAMH1 site, and this is LAC-Z prime, and we have a gene of interest, and which is also with BAMH1 sites. And now we perform ligation. After restriction digestion and ligation, what we get? What do we get? What are all the possibilities is what you should think of. And then there is always a possibility of vector-vector religation, right? That is BAMH1. That is, I'll just put it as VV. There is also a possibility of, yes, let me put it, where we get the recombinant. That means that is where this is the recombinant, and that is what we are looking for with the gene of interest. Okay, this is the fragment. We'll also get several of those, maybe multimers of fragments like this. Right? Now we will transform all these into E. coli cells. And we should choose a kind of E. coli which is the mutant. LAC-Z, I said mutant, right? Which means that it does not have this part, only the other part, which will produce only half of the beta-galactosidase. So if we transform, what are all the possibilities? We will get a majority of non-transformants, which means that they do not code for any, they did not get any bacteria at all, any DNA at all. Those are non-transformants. And then we will also get transformants. One second, I'll put it here. So assume that these are non-transformants, and we will also get a lot of transformants. Right? In the transformants, how many types do we have? Transformants are those that got some DNA. We may get those that are vector religation. We may get fragment religation. We may also get the recombinants. So we can forget about this. And what we will, if we grew these cells on plates containing ampicillin, then only recombinants and vector will grow. Vector religated ones will grow because they have the beta-lactamase gene, which comes from the vector. And we need to be able to distinguish these two. How will we characterize them? Is by adding them on plates containing. The plate should contain nutrient agar or LB agar. It should contain ampicillin. It should also contain XGAL. Now, which ones will give blue color and which ones will be in white in color? VV, say for example, in this case, I'm trying to draw here. If there is vector, and vector will produce the alpha component, and these two will form a functional protein and will convert XGAL to blue color.
So the recombinant ones are the one which gives white in color, while the ones which are not recombinant, the vector aligned are blue in color.
That's right. You're right. So if I forgot to mention, the LAC-Z gene has the multiple cloning site. So there is another term that I needed to explain that is called as, one second, that is called as insertional inactivation. What is insertional inactivation? If you saw in
Yes, sir. If the restriction endonucleases cleave the one of those selectable markers, then there's a chance that the resistance gene is lost, I think.
That's right. That's right. So if there is an open reading frame like this, which produces a gene, produces a protein ultimately, and that is a functional protein, right? It has a function. But if we have inserted some other genetic elements into it, say something has got inserted into it, now the protein may not be produced, or protein may be produced that is non-functional, right? So that is where insertional inactivation. By insertion, we are inactivating the function of this gene, right? That is called as insertional inactivation. So if we see that way, in the recombinant, what would have happened? This is the LAC-Z gene, LAC-Z prime. Because the fragment is inserted, it can no longer produce the alpha part of the beta gal. Therefore, it does not give a functional protein. That means that bacterial cell in which the recombinant is present, recombinant will remain white in color, whereas the vector religation 1 or non-recombinant will give blue color. Just by looking at the plates, at the colonies, the color of the colonies, we can recognize which one is recombinant and which one is non-recombinant. And then it's easy for us to just go ahead and perform the additional experiments. When the time it was made, it is a huge hit, PUC18. And so we don't need these kinds of old techniques now. Why are we talking about all these things? One should remember that it's not about, there are two things I will thought of covering more, but I will take the other parts later. But I want to have a small discussion so that you will understand what are the goals of why we are studying this. So there are two things. One is knowledge, and knowledge eventually gives rise to intelligence. Right? When we are learning the knowledge, if we are learning the logic part of it, then we are improving our intelligence. The ability to apply knowledge is probably intelligence. How fast you do, how resourceful one is, how fail-proof it is, how spontaneous it is. These are all the properties of intelligence. Okay, it is eventually the application of knowledge. But how fast, how fail-proof, how safe, or how spontaneous, how much low cost, right? All these things will matter for intelligence. And when we are reading about anything, we should try to know the logic of how that knowledge has come from. Because knowledge eventually gives rise to more knowledge, isn't it? Sorry, intelligence gives rise to more knowledge. Intelligence is also kind of part of the knowledge itself. When we know about the designs, we are trying to learn about principles. And that is where everything comes to life as an educator or as a learner. And therefore, we will learn about the old or little bit outdated also, because we have to see the beauty of their intelligence or their design. When we learn about logic, when we learn about design, that is when we are actually learning the intelligence directly. Because the intelligence can be applied to many other areas as well. The more and more principles you learn, the better you're getting every day. That's why I think we have to read these old ones, because at the time when they are doing it, it's a fantastic way they have done, and so it'll be nice to walk in their shoes. So I want to leave you with this one concept that we have discussed about lytic and lysogenic previously, when I was discussing about the discovery of restriction endonucleases. I was talking about it. There are many types of vectors, and the vectors can be classified mostly into plasmids. They can be based on
phages,
right? They
plasmid.
can be, yes, we will come. I'm just looking at from where they have come from. Plasmids are a good source of vectors. Bacterial chromosomes. You can also have yeast chromosomes. And people have manipulated plasmids, phages, bacterial chromosomes, yeast chromosomes, and so on to create more types of vectors. These are the sources of vectors. And then they have made cosmids, plasmids, phagemids, and bacterial artificial chromosomes. Phage artificial chromosome also they call it. One of the thing is there. And yeast artificial chromosomes. Okay. So I hope you know we have already covered the purposes or the general biology of plasmids. And we have to cover about phage as well. Yes, please.
Sir, even we can include animal and plant vectors.
Yes, they are also included. They all have come from either of these, phage, or I should have put maybe viruses. Okay?
Yes, sir.
Yeah, they will fall into one of the category. The host organism in which the manipulation is done is a different
like even plasmids, like it's also even some plant or animal vectors might be from plasmids. So yes, sir.
Yes, that's true. Yes, they will fall into plasmid categories.
Yes, sir.
Yes, that's right. So in all the cases, I think we should learn a little bit of biology before we move ahead, just like before we just go into the knowledge part of it. Okay, so I want you to have a look at these. I want you to also have a look at lysogeny and lytic cycle. And tomorrow we are going to discuss about lambda DNA, and it will a little bit of about lambda DNA and eventually the lambda vectors. I will stop until here. I will discuss about EMBL3A. Please have a look at it if possible. And this will be part of the coming CA exam until EMBL3A. Okay, today is Thursday, Friday. Right? Okay, any questions or other things? Otherwise, I would just want to, if you give me time, I will want to describe these two. I think we already discussed about lysogeny and what is meant by a prophage. If the phage integrates into the bacterial chromosome, and this is the whole of phage DNA, that is called as prophage. If it is a virus integrated into, say, human genome, then you call it as a provirus, something like that, which is usually a reversible process by site-specific recombination. And there is also lytic phage, lytic cycle, where the phage will grow, multiply, phage replication occurs, it breaks open the cell, more are released, and they will infect further many more. So that is the lytic and lysogenic cycles of lambda. The lambda DNA, when it is packaged into the host, into its head particles, it is double-stranded DNA with the ends having a 12-base overhang like this, which are referred to as cohesive ends. And upon integration into the, upon insertion into the cell. So the lambda phage sits here, attaches, and injects the DNA into the cell. In the cell, these two are complementary. So they will circularize and form a double-stranded DNA like this. It can undergo replication, or there is another possibility is getting integrated into the host genome to form a prophage. These are some of the possibilities. And if it is not undergoing the prophage, when it's not integrating and it's not undergoing lysogenic, in the lytic cycle, it will form a small nick. Sorry, okay, here is the linear DNA. These are the cos sites. They will get ligated, and the host ligase will seal it up. And then theta mode of replication will happen. You see that there is one replication fork there, and here is the another replication fork. So it can divide multiple times, making more copies of the lambda phage DNA. Then it shifts from theta mode of replication to rolling circle model. In the rolling circle model, this keeps on rotating this way, and here is a replication fork. It will be synthesizing DNA on both the strands. And what happens here is that you see from here, this is a cos site. These are the cohesive ends. When they fuse together, you call that as cos site. From cos site to cos site and another cos site. You'll have a long DNA molecule with a lot of cos sites like these, and each one is a lambda genome. This is a lambda genome, and more will keep on coming, and they are lambda genomes joined side by side, or they're produced that way. Those are referred to as concatenamers. Okay, and maybe I'll stop here. I think we are losing time. In the next class, I will describe. I'll review these and repeat and finish the lambda DNA vectors. Okay. Thank you very much. Any questions? Okay, then.
For now, no, sir. If
Yes,
anything, we'll let you know, sir.
Yes, please. And for CAA, please send me a list of topics that you want to review. But otherwise, maybe I'll give you some sample questions, and you might want to practice them. Okay? Take care and have lots of fun.
Thank you, sir.
Yes, yeah. Bye-bye.